Hi everyone, thanks for coming. It's a great pleasure today to introduce Annette Jacobsen, who's giving her PhD completion seminar today. But first of all, I'd like to pay my respects to the traditional owners of the land, the Wurundjeri people of the Kulin Nation, pay my respects to elders past, present and emerging, and other Aboriginal and Torres Strait Islanders who may be present today. Annette joined us in 2015 for honours, and so we already knew she was an impressive character then. She'd been working full-time doing her undergrad and excelled. And she'd done an internship in Sweden, and so we knew that she was lab ready. And we certainly weren't disappointed. I should say we being John Silk and I. We certainly weren't disappointed. She had a stellar honours and published a paper during that time, which is now cited to around 50 times, I think. So it's quite an impressive piece of work for an honours student, and really took that momentum into her PhD. And we're going to hear a lot about different techniques that she's developed in the lab, and she's really been a vital cog of our team because those of you who know me well realise I don't do a lot of cell culture, whereas Annette is a total gun, and she's developed a lot of the methodologies we use in the lab and a lot of the reagents that we're really reliant on. I guess without any further ado, we welcome Annette.
All right, thank you very much, James. So firstly, before I start, I just want to apologise to anyone who is coming here expecting a seminar about intestinal crypts and organoids. This is actually like crypts as in death crypts, so it'll be all about necroptosis and the death effector MLKL. So as James said, this is my PhD completion seminar. So what I'll go over today is just a little bit of background on cell death in general, then focus on necroptosis and the pseudokinase MLKL, which is our lab's favourite protein. Go through what my research question and aims were, and then go through some of the results that I've found, which, as James said, includes one methods section, which is about a non-integrating lentiviral system for CRISPR-Cas9, and then looking at the functional effects of mutations on human MLKL, and also investigating a novel inhibitor, and that should be of necroptosis. So hopefully this mistake in my first slide is the mistake in all my slides. So it's not an inhibitor of MLKL, so apologies for getting anyone's hopes up there. All right, so cell death is a really important part of life, and it's also a really important part of life here at WEHI. So when I first came to WEHI, I was in a division that no longer exists anymore, so it's cell signalling and cell death. And I thought that the 4 labs there were pretty much all of the cell death research that's going on at WEHI. But now after 4 years of being here, I know better. And this is just a selection of some of the scientists who are classed as, by the Institute, as cell death scientists, or have projects that focus solely on cell death, at least one project that focuses solely on cell death. And these researchers, over 20 researchers here, this covers pretty much all the new themes in our institute. So cell death's obviously involved in healthy development and ageing. So when in embryonic development, you need cells to die at the right time in order to get a correctly developed embryo, and when you're ageing, it's important to try and, I guess that's where your cells start to die, and that's part of the ageing process. It's also in our theme, which is infection, inflammation and immunity. Cell death plays a big part in cancer research and treatments, and because of the influence of cell death in disease, new medicines and advanced technologies also spend a bit of time looking at cell death. So there's quite a lot of ways that cells can die. I think when I was looking at the latest nomenclature on cell death paper, there's like about 18 or 19 different ways I've now categorised as cell death, but these are probably the 3 that we would most look at the Institute here. So what most people would have heard of is apoptosis. These are all regulated forms of cell death, which means that the cell gets a signal and then it undergoes a chain of events that cause it to die. So apoptosis can pretty much be performed by all cell types. It's a caspase-dependent form of cell death with low immunogenicity. So what you see in apoptosis, the characteristic signals is nuclear condensation. You get the membrane blebbing, apoptotic bodies which are phagocytosed by other cells, immune cells. So these two forms of cell death, pyroptosis and necroptosis, have got quite a few similarities in that they're both inflammatory forms of cell death. So they both involve sort of membrane swelling, destabilisation of the membrane and the release of intracellular contents or DAMPs. So pyroptosis is a caspase-dependent form of cell death as well. It's slightly different caspases, well, very different caspases to what's involved in apoptosis, whereas necroptosis, which is the form of cell death I'm most interested in, is caspase independent. And necroptosis is activated by extracellular receptors when caspases are compromised. And as I said before, it's an inflammatory form of cell death, so it can have consequences when it happens. And why we're interested in necroptosis is because it's a relatively new form of cell death that's really only been well characterised since about 2009. And even more, like some of the key effector MLKL, which we're interested in, wasn't introduced until 2012. But since that time, necroptosis has been popping up in a number of different diseases, and these are mostly inflammatory diseases, so autoimmune diseases and neurodegenerative diseases. And if you think of the fact that during necroptosis that the cell swells up, the membrane breaks and the cell lyses, these sort of diseases fit in well with the pathology of these. The pathology of these diseases fits in well with what you'd expect from necroptotic cell death. Necroptosis has also been implicated in some cancers, and there's a bit of a mixed bag with this. So most of the research tends to focus on necroptosis. Like if you lose necroptosis, that's a detrimental for you as far as cancer goes, because your cell's avoiding cell death. So resisting cell death is one of the key hallmarks of cancer. But there's also some evidence that in certain cancers, if you lose necroptosis, that it's an advantage for you. So depending on the type of cancer and the sort of signalling it's got going on, it could go either way with cancer as well. Now, because with resisting cell death being a hallmark of cancer and a lot of cancer cells resist apoptosis, inducing necroptosis has also come up as a strategy for treatment of cancers as well. And there's quite a lot of labs, this is just a small selection, there's quite a lot of labs around the world that are investigating this strategy of inducing necroptosis as a way to treat different forms of cancer. So as James mentioned at the start, I'm a cell biologist. I'm mostly interested in what goes on inside the cells and how the signalling occurs in the cells and how we can use that to better understand what's happening and I guess eventually get better treatments for the diseases like on the previous screen and possibly for things like cancers. So the type of necroptosis that I'm most interested in is necroptosis that's mediated through the TNF receptor 1. So TNF's an inflammatory cytokine, and when it binds to the TNF receptor in normal circumstances, it initiates a chain of events that involves the phosphorylation of receptor-interacting protein kinase 1 by a cellular inhibitor of apoptosis proteins. And this leads to a transcriptional response within the cell through NF-kappa B that's both pro-survival and pro-inflammatory. Now, there's certain situations within the cell that these CIAPs may be compromised. If they are no longer working, then this receptor-interacting protease kinase 1, RIPK1, does not get ubiquitinated. It's able to disengage from the plasma membrane to the intracellular space. It associates with caspase 8 along with a lot of other proteins. And here caspase 8 can cleave and prevent further RIPK1 activity and also promote apoptosis within the cell. If caspase 8 is also inhibited, then this switches the phenotype from apoptosis. So RIP1 cleavage is prevented. It can now associate it with its partner kinase, RIPK3. RIPK3 then phosphorylates the pseudokinase MLKL. And following that, MLKL undergoes a conformational change, and we believe it undergoes a conformational change and then oligomerises and translocates to the membrane, initiating cell death via necroptosis. So in this scenario, the three key interactors that I'm going to talk about a little bit today are RIPK1, RIPK3, so they're both kinases, and MLKL, the pseudokinase. We've also, there's some other proteins that are now fairly established as being interactors of MLKL, so who play supporting roles. So there's the HSP90 proteins. This was found by us here. This is part of my honours project, and also another couple of labs at a similar time found the same thing. So this actually associates with all three, MLKL, RIP3 and RIP1, and escort proteins at the cell membrane show some association with MLKL by a couple of groups as well. So there's still a few things we don't know. So we don't know exactly how MLKL destabilises the membrane, and that's something a number of groups are working on. And also whether there's any other essential protein interactions required, particularly during this process from when MLKL is phosphorylated by RIP3 and when it goes up to the membrane and destabilises the membrane. So we're pretty sure there's going to be other things involved in this process, but as yet we're still trying to firm that out. So MLKL, as I said before, it's found to be a key effector of necroptosis in 2012, and this was by two independent groups. And the first solved structure was published in 2013, and this was from our institute here by James and Peter. And this is the mouse MLKL. So far, all these MLKLs that we've seen, we think have got a similar basic structure. So there's an end-terminal 4-helical bundle domain, and this we think is this is what we know as the killing domain. So this associates with the membranes and causes the disruption. So there's the pseudokinase domain, which is the regulatory domain, and this is where the kinase RIP3 phosphorylates the protein. And in between this, there's a brace region, and that consists of 2 helices. And what we think happens with this is when the phosphorylation event happens, a conformational change occurs that causes a shift through these brace regions, releases this 4-helical bundle here and allows cell death. So the key things I probably want you to remember about MLKL for the rest of the talk. So the helical bundle domain is the killing domain. So we think, again, like I said before, this is a switch mechanism, whereas inactive MLKL before phosphorylation is constrained, and after phosphorylation, the helical bundle is released to be able to cause cell death. So also, MLKL forms oligomers, and there's been quite a number of groups who've shown this as well, and translocates to the membranes as well. So these are established facts. And MLKL is a pseudokinase, and also, I just wanted to stress in here, before I go on a bit more about what a pseudokinase actually is, that most of our knowledge of MLKL function actually comes from studying the mouse protein, and there's a few reasons for that, but I'll go into a bit later. All right, so a pseudokinase, it's a protein that has a kinase-like domain, but it's got either impaired or completely absent catalytic activity, so it can't phosphorylate a substrate. So in a normal protein kinase, what it needs to be able to do, it needs to be able to bind and coordinate ATP and essential metal cofactors. And these motifs I'll talk about a bit later as well, but these are sequences of amino acids within the protein that are required to do these activities. Also, the protein needs to have sufficient catalytic activity to transfer the phosphate off ADP and put it onto the substrate. And finally, the protein needs to be structurally capable. So if it's not the right shape to do what it needs to do, then it's not going to happen. So a typical protein kinase will have a C lobe and an N lobe, and the way these two interact and move together will determine how it works. If any of these four things is out of place, and sometimes in pseudokinases you'll find more than one of these things out of place, then the protein's classified as a pseudokinase rather than an active kinase. So just before I go on to what my research has been about while I've been here, I just wanted to just quickly touch on some differences between mouse and human MLKL. So we've got some differences in tools. So the structure that I showed before was for the mouse protein, which we've had for some time, but we've got no full-length human crystal structure as well. So this means we have to make some assumptions about what the protein looks like and how it's going to behave when we manipulate it. We've also had some difficulties getting, at least in our lab, had some difficulties getting reconstitutable knockouts in human cell lines. And because the MLKL knockout mice have been available for some time and they're quite healthy and they're fertile, so they breed, that we've tended to do most of our research in mouse cell lines because of the availability of them. So the issue we did have with the reconstitutable knockouts was with the CRISPR-Cas9 guide expression. So when we introduce the guide RNA into the cell, I'm not going to go through CRISPR too much because this is a technology that most people know, but if you don't, it's a technique that we use to modify genes within a cell. So the guide RNA, what we find is when we have that inside the cell, even if we have an inducible expression, so even if we turn it on command, there's still enough expression of this guide RNA when it's turned off that prevents us from putting the endogenous protein back into the cell. And this means that we can't reliably assess the effects of mutations without the presence of the normal MLKL there. There's also a number of differences in function that we've noted so far between the mouse and the human protein. So we've got a phosphomimetic mutation in kinases. Their phosphorylation is related to their activity. And what we've found is when we mimic the phosphorylation of the protein in mouse, this causes an autoactive protein that kills in both mouse and human. However, in the human protein, this doesn't seem to happen. I've put a question mark down for all the human ones here is because these were all tested with the endogenous MLKL in the background, so the wild-type MLKL, so it wasn't a completely clean system. But it appeared that the things that were active in mouse were not active in humans. So you can see like where the mouse is all green, the human's all red. So they behaved quite differently. So in the human protein, the only way that we were able to see activity without involving the upstream kinase RIP3 was to force the dimerization of the N-terminal killing domain. So because MLKL oligomerizes, if we bring those two killing domains together, even if the pseudokinase domain is not there, that can cause the cell to die without any other activation. So the two main research questions that I wanted to address during my PhD was which region of the human MLKL proteins are important for function, and how are these similar to or different from the mouse protein? And this was sort of a broad research question. There was originally a couple of projects involved with this, but the question that we were asking broadly is whether there's still novel essential effectors of MLKL downstream of MLKL activation. And this kinase inhibitor came up in a screen from the Institute. So I was specifically investigating this kinase inhibitor to see if we could identify a necroptosis effector through researching this drug. So the three aims that I'm going to go through. So the first is to establish a CRISPR mediated gene knockout using non-integrating lentiviral systems, then the function of various MLKL mutants in the absence of endogenous MLKL. So aim 2 relies very much on aim 1. And finally, I'll just take you through a brief overview of what I found out about the kinase inhibitor AMG 47A. Right. All right, so before we go through, so this part of the talk is all about developing tools and making sure you have the right tools. I guess the first thing I want to say, we've got different methods of getting the CRISPR-Cas9 system into the cell. And when we're considering what way to do this, so you've got a number of non-viral vectors, but these involve physical and chemical methods, which can be quite harsh on the cells. So what I like to do with that is look, because we use viral vectors quite a lot in the cell, and they're quite gentle, and they can be applied to pretty much all cell types. So we decided to focus on doing CRISPR delivery via a viral vector. So the type of vector we use are called lentiviral vectors, and these are really versatile because they can infect both dividing and non-dividing cell types. And they can pretty much, they work really well in cell culture, which is what I do. And they can pretty much infect all the cell types that we use, and we use them regularly. So what happens with this is that once we have the vector that's introduced into the cell by the viral construct, the viral genome, which is, well, the proviral genome, which is made of the RNA, which is made of RNA, is transferred to DNA by reverse transcription, and then it's integrated, formed into a pre-integration complex, which is taken into the nucleus, integrated into the genome, and then you can get protein expression of the gene that you've decided to introduce in the viral packaging. So the integrase protein, most of the lentiviral vectors that we use are based on HIV. So it's a way of like taking something that can be pretty nasty and finding a really good use for it. And the HIV-based vectors are, as far as my work goes, they've been essential throughout my PhD, and it's been the way that I found out a lot of my work. So, the protein that I was interested in when we're looking at doing a virus that doesn't integrate, so it doesn't do this step, is a protein that's aptly called integrase. And this does a number of things. It binds and processes viral DNA, as well as host DNA, and it also catalyses the integration into the host genome. And there's a number of different ways that you can mutate integrase so it doesn't work. So you can mutate some of the binding sites that bind to the DNA areas, but what's most commonly done is that one of these three catalytic residues in the core catalytic domain are mutated. And the one that's most commonly done is this aspartate 64, which is the first out of the catalytic triad. All right, so for the generation of the non-integrating lentiviral system, I used two different delivery vectors. So we used our standard PCMV delta 8.2 to introduce a D64 mutation into that by overlap mutagenesis PCR. So that meant that I could compare what we used normally in the lab with the non-integrating system to see the change in function. And I also purchased this plasmid as well, which is from Adgene, and it still is to this date the only D64 mutant packaging vector in the whole Adgene database, and the only citation of this vector, at least through Adgene, is our paper. So it hasn't been used that much, from what I can tell, although there are a lot of commercial systems available to do this sort of thing as well. So why this works is because when the DNA is in the nucleus, you do, like in the normal systems when integrase is active, the majority of the viral DNA is in this linear double-stranded DNA form, which is then integrated into the host genome. But if you block this integration step, what you find is that this linear DNA, it's more likely to go through different types of recombination, so non-homologous end joining or homologous recombination, and form these circular episomes, which can then be transcribed and translated in the nucleus without going into the host DNA. And these episomes, because they're not integrated, when the cells divide, it will just gradually dilute out of the cell. All right, so the first cell line I tested this in, and seeing that I'm talking about human mutations is a bit weird that I've got mouse up here, but things changed during a PhD, and when I started doing this project, there was going to be something more mouse-based with CRISPR. I guess what you can see here is I've got the integrating virus compared to the non-integrating virus. So there's two non-integrating and one integrating, and then a control which doesn't have anything in it. And when I was first testing this, I was doing this at 2 different temperatures because after doing some reading, there was some suggestion that if you drop the temperature during the early stages of infection, that you can prolong the infection and I guess have a better chance of your desired effect happening. And this is because the half-life of a lentivirus at 37 degrees, which is what we normally cell culture at, is about 6 to 8 hours, whereas at about 30 degrees, it's 24 hours. All right, so as you can see with these, if you look at the non-integrating ones at 30 degrees, so that's the brown and the green, but they're much higher than the ones that are at, the brown at 37 and the green, so they're from the purchase one at Adgene, and the green one is much higher than the brown one, so we are maintaining the vector for longer. And that's the same with the one I made, except the effect wasn't as good. The next step with that was to just optimise a spin technique to try and get more virus titer. So when you have a non-integrating lentivirus, there's a couple of disadvantages you have, and the first is that not as much virus is produced in the first place. When you get the virus into the cell, not as much product is transcribed as well. So to maximise the amount of virus we're getting into the cell, so I used a different spin technique to concentrate the virus down into a small amount and then resuspend it. And so this was done at relatively high centrifugation speed with a sucrose cushion, so that protects the virus. So as it sinks down during the centrifugation process, the viral capsids are not damaged and you get better infection efficiency. So you can see with this that the ones that are at 30 degrees pretty much maintain the level of virus within the cell, so it all stays up high, whereas the ones that are at 37 degrees, so that's the brown one and this one down here, the dark blue, they drop down over cell division. And this is sort of what it looks like compared to a normal cell, which has the fluorescence down really low. The integrating one is actually quite high, and even though these are still pretty much all over the line, you can see that there is a drop in the amount of the intensity of the fluorescence and then relatively the amount of protein that's expressed. So the first cells that I tested this in again were MDFs. So I thought it should try an actual knockout, and I used a RIPK1 guide that was designed by Denise Heckman because it's a really good guide. So you can see if you've got an integrating system here, and this is a pool of cells, that it pretty much knocks out everything. And I was encouraged to see that using the non-integrating system, compared to wild type, you were able to still get an effective knockdown of the cells, although it wasn't as effective as when it was integrated, which is what you would expect, but it was good to see that it was working. So the testing then in human cell lines, so again I tested both the vectors at this stage because, look back here, so the difference between the amount of knockout between 30 degrees, which is this one here, and the 37 degrees is not that much, and if anything, it's even a bit better at 37. So even though you're keeping the level of virus in the cells for longer, the lower temperature is probably affecting the transcription of the gene product anyway. So, and the transcription of a guide RNA anyway. So it didn't seem to be any advantage and just took more time. All right, so in the human cells, both the vectors behave pretty much similarly. So the black and the orange up the top is in the U937 cell lines. That's a human cell line that we use quite regularly. And the red and the green one here are in a different human cell line, the HT29, which are a colonic adenocarcinoma cell line. So as you can see with this, the peak of the fluorescence comes in at about 2 days. And I did adjust the concentration of the virus after this to get a better effect and more initial infection. So you can see with the HT29 here that at day 2, when it was adjusted further, that pretty much all of the cells at day 2 have a high GFP fluorescence. Whereas after day 9, so that's the second sort out here, you can see that most of the cells have lost the virus, but there is still a population that does retain it integrated in. All right, so the general method that I went through here was to produce the virus in our 293T cells, then to take it out and concentrate it using the centrifugation technique I discussed earlier. So it had produced 2 sorts of this. So in the first sort, it will take all the cells that have very high levels of GFP because we know that they have a lot of the CRISPR vector inside them. And the second sort we want for the low GFP, so that's this group down here, and that's because we want the cells that have diluted the vector out. So there's the random integration that comes in here, although it might get a more effective knockout, it would still have the problem that it still has the integrated guide in there, which is what we're trying to avoid. And then there's the last process is to test for gene knockout in both pools and single cell clones using phenotype and Western blot, and finally confirm the clonal knockouts with next generation sequencing. So after all this, I have 2 groups of human knockout cells. So there's the U937 cell line and the HT29 cell lines. And these, as you can see, so this is the wild types. This is the normal level of how the U937s die. So when you induce the protein with doxycycline and then treat it with stimulus to induce necroptosis, you do see cell death. It's not quite as much as what you see in the wild type cells, but overall I tend to get a little bit less protein expression than endogenous in the U937 cell lines. With the HT29 cell lines, so this is the proteins with the normal wild type MLKL, and these are the reconstituted cell lines, so they only die with induction of the protein with doxycycline and a necroptotic stimulus, and that's about at an even level with the wild type cells. All right, so with that done, so we've got a system that's developed to do non-integrating lentiviral CRISPR it's validated in mouse and human cell lines. Reconstitutable MLKL knockout cell lines were developed. Since then, I've also got reconstitutable RIP3 HT29 cell lines, and we're also testing the system with a dual Cas9 guide RNA vector. So hopefully that eventually the cells will have no guide in them and no CRISPR, no Cas9, so no nuclease in them as well, which would be great. All right, so the next section I want to talk about was the functional effects of mutations on the human MLKL protein. And most of this data is published in Petri and Sando et al. So if you want more of a story, that is the place to check it out. So I'm just going to talk about a couple of these mutations today. So the first thing I just wanted to highlight is that although MLKL is a pseudokinase, if you look at it next to a canonical protein kinase like MAP3 kinase 1, you can see that it still has all the structural and, I guess, functional, well, it's got structural similarities. All the domains are in the right place, the N-terminal lobes. Like it's recognisably a kinase is, I guess, what I'm trying to say. And the key catalytic motifs that you find in canonical protein kinase are still here in MLKL, except some of the residues you can see are different, and this is what makes it unable to catalyse a reaction. So the first group of mutations I wanted to talk about are the effects of cancer-associated mutations on MLKL function. Now, cancer-associated mutations are not found very often in MLKL, but the ones that do, they all appear, well, the ones that we were interested in, I don't say all of them, the ones that we were particularly interested in were the ones that were around the catalytic motifs or the pseudo-catalytic motifs of MLKL. And this included, like in some of the catalytic motifs itself, in the case of the E351 and G330. And for the E258, it sits right in between. So it's in a similar sort of area. So before I go on to what I found, I'm just going to very quickly brush over what Emma and Jared found about the difference in function between this protein and the wild-type protein. All right, so firstly, when Emma was purifying this protein, so normally, so for these graphs, all the top is the normal MLKL, and the bottom is the E351K mutant. So when you normally purify MLKL, you get two distinct species, so one at a higher molecular weight and one at a low molecular weight. But with this protein, we only got the one species at the lower molecular weight. And this was verified again by microfinite Bio21 with analytical ultracentrifugation. And again, so these are in the opposite order here. So you get a low molecular weight and a high molecular weight species appearing for wild-type MLKL. But with the E351, you only get the low molecular weight species appearing. And Jared Sandow checked out this species here in the wild type and this one here in the E351 and confirmed that this here was tetrameric in nature, whereas this was monomeric in nature and some of it was dimeric. So what they found with that was that this mutation does impair the ability of MLKL to form tetramers. As far as the function goes, then Emma tested these in liposome assays. So this is where you have a self-quenching dye called carboxyfluorescent. And this, when you package it into lipids, when they're all close together, they stop each other from having fluorescence. And if you treat them with a disrupting compound, or in this case, disrupting protein, causing the dye to leak out, then you can measure the fluorescence and you can get an idea of how many of these liposome structures are being disrupted. So with the wild type MLKL, so this had clearly two states. So in the presence of ATP here and with the E351, so they behave the same in the presence of ATP. However, when ATP was taken away, the wild type protein busts up these liposomes a lot quicker, whereas the E351 is a little bit impaired in the function, so it's a slower disruption of the liposomes. And the other cancer-causing other cancer-associated mutations all showed a loss of responsiveness to ATP as well, and they all stayed at a similar sort of rate at what the wild type with ATP was doing. So they never achieved this faster rate of liposome disruption. So when I looked at these inside cells, so it's this N column again that is the one that I want you guys to focus on. So this is the wild type, and it's going at about 80% cell death. And the black column is 6, and then you've got 12 and 24 hours. So we can see with all of these three mutants, the E351, the E258 and the G330, that they are all impaired at the early time points, and the G330 is also impaired up to 24 hours compared to the wild-type cell line. So these mutations all affect the function of MLKL inside cells. And this was also, so in the Blue Native page, what we see with a wild-type MLKL, when you treat it with a necroptotic stimulus, that you get these characteristic high molecular weight functions. You can see this in the reconstituted cell lines, but again, there's a delay in these complexes forming in cells expressing these cancer-associated mutations. So the other mutation I just wanted to talk about here was the lysine 230. So that's part of the canonical kinase Vake motif, which is responsible for coordinating ATP. And we know that MLKL does bind ATP. And why I'm including this one in here is that this protein has been reported to be mutated in cancer, although it's not the exact mutation that we tested here. So again, looking at the end one here, you can see that this version of MLKL is less effective than the wild-type protein. And this is also shown in the, well, it's also in the liposome assays. You can see that there's a similarity with the other cancer-associated mutations, and it no longer responds to ATP. And this is also interesting as well, because in the mouse protein, we see quite a different thing. If you mutate the equivalent residue in mouse, you actually get an overactive protein that causes cell death without needing any stimulation, necroptotic stimulation. All right, so the next group of mutations of MLKL I want to go through is the phosphorylation site. So there's two key sites on human MLKL that are known to be phosphorylated by RIP3. And there's two different types of mutations that I looked at here. And one is a phosphomimetic, and that's where we change the residue from serine or threonine to large negatively charged residue to mimic phosphorylation. And the other is ablating, where we change it to alanine to prevent any phosphorylation and to mimic, I guess, the non-phosphorylated state. So these mutations have been investigated in previous studies, but not in a knockout background. What Maria found when she was looking at this in wild-type U937s was that when she induced the protein, it did look like it was decreasing the amount of necroptosis that was occurring. Although because the endogenous protein was still there, it's hard to definitively say what is going on, but it is looking like it's acting as a dominant negative in this circumstance here. And again, this is different from what you see in the mouse, because the phosphomimetic version in the mouse causes an overactive protein as well. So the two I'm going to show you today, so I did do the single mutants for all of these, but they all behave like wild type. So the two I'm going to show you are the double phosphoablating and the double phosphomimetic. And again, it's the last box in the end, comparing to the wild type, the last box in the end. And here you can see that these mutations, whether they're phosphoablating or phosphomimetic, they completely inhibit the ability of MLKL to cause cell death. All right, and again, with the phosphomimetic, you don't see any high molecular weight complex forming here, even though that the protein is expressed in sufficient amounts. Two final mutations I'm going to talk about are the F386A. So this is located in the C lobe of the kinase, and it's normally thought of the pseudokinase domain, and it's normally thought of as a substrate interaction site. So this as well, if you look here, again, comparing wild type to here, it completely restricts the ability of MLKL to undergo necroptosis. So likely this is binding some interactor. And the second mutation is it's a double mutant D107A, E111A, and this actually occurs in the 4 helical bundle domain, so in the third helix of that. And this one as well also completely blocks MLKL activity, but what is really interesting about this one is it's still phosphorylated by RIP3, and it still forms these high molecular weight complexes, although you see the complexes forming quite evenly in both the cytoplasm and the membrane, which is not typical from what we see in normal progression of necroptosis. And they don't cause cell death, which is, it's an unusual situation that we see a protein that can be phosphorylated, can form a high molecular weight complexes, can go to the membrane, but doesn't induce cell death. All right, so the impaired functional effects, so there's a number of mutations in cancers that you see impaired functional effects on MLKL activity, and this highlights the importance of the pseudoactive site of MLKL for its activity as well. So even though it's not a kinase, this region of the protein is still really important for its function. So just verified that the phosphomimetic mutations don't cause autoactivity in human MLKL, and identified both the F386A and the D107A and the E111A as loss of function mutants of human MLKL as well. Now, the very last thing I want to go through is an abridged version of the AMG 47A story. And so I guess this was originally, this screen was originally designed set up and performed by James, Guillaume, David, Mark and Kim. And it was designed to identify, for two reasons, I guess, to see if we could identify novel inhibitors of MLKL, and also if we could identify necroptosis effectors downstream of MLKL as well. So it had two systems there. So there was a stimulus-dependent screen that was performed on, I guess, cells that had been treated with a necroptotic stimulus, which was a SMAC mimetic to block the CIAP activity, a caspase inhibitor to block the caspase activity, and TNF to push the signalling pathway to get the phosphorylation of RIP3, of MLKL by RIP3. And then there was also stimulus independent, and this was using an activated MLKL mutant that could cause cell death independent of the upstream signalling RIP1 and RIP3. And this screen was set up a while ago, so this is all done in mouse. And the mutant that was used is the Q343A mutant, which was shown by James, Peter and Joe, that it also caused cell death just on expression alone. It didn't require the necroptotic stimulus to cause cell death. All right, so this screen was tested with 438 compounds, and they all had a known mechanism of action, which is important because once they were identified, we sort of knew what direction we were looking in for, I guess, what the target could be and what the effectors could be. All right, so this compound, AMG 47A, kinase inhibitor, it came up in both screens. So it showed protection from TSQ-induced necroptosis, and it showed some protection from cell death caused by the Q343A mutant. And this AMG 47A, it was originally developed as a lymphocyte-specific kinase inhibitor. So it's this compound 47, and it inhibits lymphocyte-specific kinase at sub-nanomolar levels, although it also inhibits a range of other kinases at nanomolar levels as well, including other Sark family kinases, MAP kinases, VEGFR2, and some of the JAK kinases as well. So the first thing I wanted to do with this was test in human cells, and what I found was quite nice. That was it very robustly inhibited necroptosis in the U937 cell line. So that was great. So I went and tested in a number of other cell lines and found that in all the cell lines I tested that this drug was able to protect the cells from necroptosis. So that included HT29, the THP1 as well. So then wanted to verify the screening results in mouse cell lines, and here's where it got a little bit frustrating. It was at the time. And in this case that I found that I couldn't replicate the results of the screen, and I didn't see any protection in the mouse dermal fibroblasts, which were the cell line we used in the screen. That testing with the PI, it didn't appear to have any protection from cell death. So because this was a little bit unexpected, we checked this in a number of different cell lines as well in mouse, and found that there was some protection in raw macrophages, and there was some protection in some AMLs, but not others. So it was a little bit of a confusing story for the mouse here. But what we also noticed was that the mouse cell lines had quite high toxic responses to this kinase inhibitor. So at the doses where we were seeing effects, there was also quite a bit of associated toxicity. So we looked across in a panel of different cell lines that are involved in necroptosis. Well, these are knockout cell lines that we have available from mice that are involved in necroptosis, extrinsic apoptosis and intrinsic apoptosis. And the only cell line that we found that could be protected from the toxic effects of this drug was the back double knockout mouse embryonic fibroblasts. So that showed us that the AMG 47A toxicity is BAX and BAC or back dependent form of cell death. So the next question was, how is this drug working in human MLKL? And originally I tested this with the dimerizable gyrase constructs of the N-terminal domain, so either the 4 helical bundle on its own or the 4 helical bundle in the brace region. And if we dimerize these with the divalent antibody, kumamycin, this causes cell death absent of upstream stimulation, and these cell lines were made by Maria Tanza originally. So the results I got from this were a little bit confusing, that I didn't really see, so looking here between the black and the red, so the black is without the drug and the red's with the drug, there wasn't really much difference in protection when I was using the drug here. There was a little bit. However, if I did the same experiment in the background of QVD, a caspase inhibitor, I actually could see quite clear protection in both the N-terminal domain gyrase and the helical bundle domain gyrase. So after I got the MLKL knockout cell lines ready for use, I regenerated these in both the wild type and the MLKL knockout U937s. And in this case, what I found here was that the MLKL knockout U937s didn't behave how I expected. So there wasn't, like, this is, so you get an equivalent amount of cell death between the wild type and the knockout, which is good. And you do get protection when you have the QVD and AMG together, which is what I found before, which is good. However, when I just used QVD to see if I could inhibit the cell death, this nearly completely blocked the cell death in the MLKL knockout cell line. So this suggested that what was happening here was more a caspase-dependent form of cell death rather than an MLKL-dependent form of cell death. Or potentially both, if that's confusing. So I ended up playing around with the methods a little bit, and I was trying the doxycycline and kumamycin simultaneously, and also doing like an overnight or over 2-night doxycycline pre-treatment, and then adding the kumamycin. And when I tried this second method, what I found here was that I got, again, an equivalent amount of cell death between the wild type and the knockout cell lines, so when I'm expressing this mutant protein. However, in this case, none of the inhibitors are blocking the cell death. So I think in the end, this is probably a more representative result of looking at the function of, I guess, whether this drug is acting downstream of MLKL activation. So from this, I presume that the drug is not acting downstream of MLKL activation. So the next step with this was then to look at the obvious suspects. So if you look in a Western blot to see if you have, I guess the main thing we'd really look for is the phosphorylated MLKL. And when you treat with a necroptotic stimulus, you get robust phosphorylated MLKL. However, when I added the AMG 47A, this completely blocked that phosphorylation. So this suggests that it's something that's acting upstream of MLKL activation. So we sent the cell, we sent the compound and off to Discoveryx to do a kinase binding assay on all the necroptosis effectors. And in this, how that works is that they have the kinases bind to an immobilised ligand on the bead, and when they add the drug, it competes them off the beads, and then you can check how much kinase has been disrupted with a DNA tag. And what we found in this is that AMG 47A actually does bind the human RIP1 and RIP3 proteins, and the KDs are in the nanomolar range for both RIP1 and RIP3. We did also test MLKL, but there was no evidence of binding to MLKL. And this is the final little bit of extra information. So this is some more data from the screening lab. So there's a RIP3 overexpression assay that was developed by Ying, and the stuff from the screening lab was performed by Amanda and Kim. And we do see some quite significant protection actually from this overexpression in U937 cells with AMG 47A, and that's as compared to NSA, so it's a good 60–70% protection. We also see a complete block of RIP kinase activity, which is equivalent to the known RIP kinase inhibitor GSK872. And although this is quite high dose, this was eventually titrated out, and it does block the kinase activity at a similar dose to GSK872, and probably even less. All right, so finally with this, so AMG 47A inhibits necroptosis in a range of human cell lines, and that's through RIP1 and RIP3 binding, and it has variable effects on necroptosis in human cell lines and has induces in mouse cell lines, and induces toxicity in a back-dependent manner. All right. It's a little bit longer than I hoped. So I guess that just leaves me to thank all the people who need to be thanked. So to my supervisors, James and John, for their support over the years. This has been a really interesting project to work on, and I've learned a lot. And you've both allowed me to, I guess, choose my own way through a lot of the PhD, which has been great for developing my independence and has given me confidence that going on to the next step after this is not going to be too big a jump. To my committee, who's always shown me a lot of support over the years, which has been really great, and also provided me with useful advice. To everyone in the Murphy laboratory, so Emma, her data you've seen a bit today. So Sam Young did pretty much all the DNA constructs that were used in that project as well. And Cherie's been helpful. So I've been doing some protein expression stuff, which wasn't presented today, but she's been really helpful in helping me with that. Also, everyone in the Silk laboratory, all the postdocs throughout the years I've been here have been really helpful along the way, and willing to give advice when needed and required. And all fellow PhD students, both in Murphy Lab and in Silk Lab, who I've shared this journey with and had companionship along the way. So Catherine Hall, she helped out with teaching me how to do the setup for the next-gen sequencing. Past division, lab and division members, particularly Maria Tanza, who, as both my supervisors are lab heads, and now both of them are division and theme heads. So Maria, at the start of my honours and PhD, pretty much acted as my supervisor on the floor, being a senior PhD student. So it was definitely like her help over the years has been great. And there's a number of other people here who are involved in the project along the way and provided support. I think I've spoken about most of the people directly. The DivCos for looking after all the paperworky type stuff and helping me navigate that side of things. All the other WeHi support staff, we're really lucky here. The choir and the inflammatones, because music is great and has been a great release. And finally, to family, friends, and all my mentors for providing support along the way as well.
Thanks, Annette. Do we have any questions? We've got maybe time for a couple. Few. I was going to kick this off. I have to show you how poor I am at throwing.
Thanks, James. Whoa, hey, feedback. Okay. Yeah, I'll try it like this. So just backing up to your sort of methodology, at the beginning you introduced the problem with the CRISPR in human cells was that the guide RNA was really potent and knocked down whatever you were putting in, I guess, as well. At that point, did you consider whether it was possible to just reconstitute MLKL with a sequence that has the guide target mutated? Is that something that was possible?
They actually did try that, and that still didn't work. So, yeah. There was still enough recognition. Well, we're presuming there was still enough recognition left that it was still causing problems.
Yeah.
I might ask, your RIP1 knockout that you showed, you still saw some residual RIP1, and I wondered if you...
I didn't show a RIP1 knockout.
Your CRISPR development.
Oh, okay, sorry, with the MDFs, yeah.
Yeah, and you still saw some residual RIP1, and I wondered if you knew the origin of the RIP1.
Oh, they were pools, so there would be some cells expressing RIP1 and some cells that weren't. So.
Anybody else? Everyone else wants to drink.
So why, with the autoactive mouse mutant, was AMG still inhibitory in that screen? Do you have any idea?
Yeah, that's like one of the things that's been bugging me for most of this project. So, because it does, like, the drug does work really well in human cells. So what, if I had to guess about what I think is happening with this, is that potentially you're getting some, like a dual cell death, you're getting some necroptosis type of cell death happening and some apoptosis type of cell death happening. And because we know this drug can induce an apoptotic form of cell death in mouse cells at quite low doses, that it's possibly switching the type of cell death over towards a more apoptotic phenotype, and possibly that is making a difference in the readout from the cell-tided glow. So you might have more ATP present, whereas with the necroptosis, if the membranes are lysed, all the ATP leaks out and that drops it really quickly. So yeah, so I think it's probably just more a change in kinetics, that you're still seeing cell death, but it's not the same sort of cell death as before.
Oh, okay.
Did you see any change on the phosphorylation of RIP1 when you treat cells with the drug? I think, if I remember correctly, there's an increase in the RIP1 phosphorylation.
Yeah, so what I've seen with RIP1's a bit funny. So it does appear to be delayed. So the start of phosphorylation is delayed. However, yeah, it is maintained for longer. And sometimes you also see, like, it looks like that there's slightly shifted molecular weight RIP1 as well. So yeah, I'm not exactly sure what's going on with that, but because the cells aren't dying, possibly that's allowing more RIP1 phosphorylation to build up from whatever activity is there. Yeah, so I'm not sure. At the moment, I think the only kinase we really know that phosphorylates RIP1 is RIP1. So if it's inhibiting, or it's binding to RIP1, I guess you would presume it's inhibiting the kinase activity, but we don't have a functional assay that's reliable enough to test that.
Thank you.
Everybody happy? Okay, thanks. Join me in thanking Annette. And we have refreshments outside.